Ahí donde lo veis, licenciado en farmacia en Santiago de Compostela, doctor en ciencias experimentales con especialización en modelización molecular y diseño de fármacos en la Pompeu Fabra en 2004, y desde el 2004 hasta el año pasado ha estado entre Galicia y Uppsala en Suecia, formando su propio grupo de investigación, alguna spin-off, y enfocado en el mecanismo de regulación de los receptores acoplados a proteínas G, que básicamente es el target de aproximadamente el 30% de los fármacos que tenemos en el mercado. Es catedrático de bioquímica computacional en Uppsala, ¿no? Y desde el año pasado científico titular del CSIC, asociado al CIP, y se vino al ISPA para formar su grupo de bioquímica computacional y bioinformática estructural. Más méritos, pues más de 110 papers, revisor, beca, supervisor de tesis.
Lo que todo el mundo que hace ciencia, vamos.
Sí, lo que están metiendo aquí para rellenar. Pero bueno, pues ahí lo tenéis. Hugo, the floor is yours.
Muchas gracias. Bueno, pues nada, es un placer estar por fin presentando lo que hacemos en el nuevo laboratorio de bioquímica computacional aquí en el ISPA. Y lo que voy a presentaros es, pues lo que Gonzalo ha dicho, ¿no? Cómo trabajamos sobre todo en receptores acoplados a proteína G desde el punto de vista de la biocomputación. Un poco, ¿quién de aquí utiliza o ha utilizado herramientas bioinformáticas a un nivel medio o avanzado? ¿Vosotros dos? ¿No? Un poquito, Miguel.
Define medio avanzado.
Bueno, algo más que utilizar un servidor web. Vale, ok. Eso es lo que me imaginaba y por eso también un poco creemos que tenemos un encaje en este instituto. Nosotros, también, a ver si tiramos para atrás. Nosotros también utilizamos secuencias, pero además de hacer un análisis de secuencias, que hacemos poquito, lo que solemos trabajar es en sacar estructuras de las proteínas que codifican estas secuencias y trabajar sobre estas estructuras. Hacemos simulaciones computacionales de esas estructuras y sobre todo intentamos modularlas, buscar cómo otras moléculas pueden interferir en la función de esta proteína, con una función obvia que es buscar nuevos fármacos. Estamos en el sótano, en el F12-1, en el pasillo hacia esta dirección al final del todo, en las mazmorras, y como decía Gonzalo, vengo de estar 12 años en Suecia, donde tengo aún contactos y tengo una pequeña carga docente y un estudiante aún. En el laboratorio está Oscar, postdoc en el grupo, y Lea, que es una estudiante de máster que visita desde Croacia. And now I will switch to English because I think it's good to make these seminars in English, especially if there's international people working, which is what we should have at our institutes. At least I know of one. So that's also what they said, what they always did in Sweden. And I appreciate it quite a lot because my Swedish sucks. So I think it's good to do this. Anyways, so I continue to describe what we do and what is our main focus. Our main focus is to look for new molecules that modulate receptors or other proteins. The chemical space is huge. This number of zeros is around 33, 30, 35. So it's huge, but still what we are targeting normally is just a very small fraction of the chemical space. So when we look for new drugs, for new medicines, we would like to explore the alternative chemical space. And for that, we work at the preclinical stage. And then most of you guys that are doing more, some preclinical, some clinical assays, then we can proceed further and the compounds would be patentable. And to do that, to explore the rest of the chemical space, we need computational tools. Now, this is possible because the computational power has grown exponentially And also, another exponential growth is shown here is the one of structural biology. We know more and more about proteins, and even more since AlphaFold and the accurate modeling, three-dimensional modeling of proteins. We have structures of proteins. Therefore, we can think on combining, oh, this is counterintuitive, so it advances on the back. Okay, so we combine analysis of big chemical databases of compounds with analysis of the structures where these compounds bind. And here you see something that most of you recognize, AI. So we also get into the AI world, and we try to analyze this huge amount of data with AI, machine learning, neural network methods, etc. At the same time, we use more classical methods on simulating how a drug binds the protein. Now, I want to tell you these methodologies in the context of the receptors that I mostly work, G protein-coupled receptors. At the end, if I have time, I will just pinpoint that we also work on enzymes. At least now you know it. We also are interested in working in enzymes, CDK, for example, CDK2 we have worked with, and other enzymes. But I want to introduce you to the world of GPCRs. This is not the first seminar of GPCRs that is given in this seminar series, but still, and how we attack this problem and how we combine it with other approaches. So just a recap, textbook recap. So GPCRs, this is weird. It should be in the background. Something was messy on the presentation when it was transferred from one to another. Anyways, so the receptor is a memory receptor that is in equilibrium between the active and the inactive conformation. The capacity of activating the receptor in this equilibrium is called basal activity. So in the absence of any stimuli, the receptor can have some basal activity. The stimuli is the agonist that binds the receptor, traps it in the active conformation, and therefore the G protein binds, and then it dimerizes, and then it goes to the downward signaling process. This is how all of them work. Now, how do we modulate it? Well, we can stimulate with a molecule that is similar to the agonist, or block it with a molecule that is similar but not too much, that blocks the agonist binding site. So these are so-called orthosteric ligands, and the classical is full agonist or neutral antagonist. We have also partial agonist when you don't reach the EC max, or the inverse agonist when you also block the basal activation of the receptor. But we also have other strategies. The allosteric modulation, like in any other enzyme, you can glue molecules in other parts than not the effective side of the molecule, and then in this way you kind of bias the signal towards a more active receptor or more inactive receptor. The pharmacological response is different, typically lower in the E max. And we also have mutations, and some of the pathogenic mutations actually act this way. And one of the goals of our currently funded project is to find some cancer-associated mutations in one of these receptors, which I will talk about, the adenosine A2B receptor. How do they hyperactivate or block the activation of the receptor? And then, last but not least, we have to consider the receptor signals either through the canonical G protein signaling or through other signaling. Can be another type of G protein or can be beta-arrestin. And the bias signaling has been associated, for example, with the beneficial effects of some drugs without less side effects. And one example is the opioids, which are now currently redesigned or rethought for being biased towards the therapeutic signaling pathway. Now, all this exists in a wide variety of receptors. We have around 900 receptors encoded in the genome, and we have a structure to many of them. We have worked in the past in angiotensin, neuropeptide Y receptors in collaboration with Novo Nordisk, and with adenosine receptors, which is what I will present today. And we have other projects, and anyone working with a GPCR, please come to us. We are interested in all of them. Now we are actually starting collaboration on olfactory receptors, thanks actually to a seminar that was presented here by Eva Martinez Pinilla. So Well, it was said 1/3 of the drugs in the market target a GPCR, but also 1/3 of the drugs in the clinical trials also target GPCRs. Now, to solve this problem, we propose solutions. So we are a computational group that not only uses methods, but also we develop our own methods. So one of them, anyone can use it. You just click on this QR code, and if things work well, even in your phone, you can see the web server GPCR ModSim, which is a web server that we developed where you plug in the sequence and it gives you a structure of the receptor. It uses AlphaFold, but it also models not only the database, but it also models the receptor if there is some mutant version in your sequence of the receptor that is not in the AlphaFold database. And it also puts the receptor into a model of the membrane and prepares it to run molecular dynamic simulations. This is some sampling that we use to interrogate this structure and learn more about the properties of this structure. And I'll tell you later a bit more about it. Now, this is how it works in the background. As I said, we use AlphaFold, but also other software, and we have like a kind of maintenance here. I will try to maintain it with a limited funding, but that you don't see, you don't need it. And we put this in the context of a pipeline to develop new compounds for GPCRs. This is the computational part, and we also, as Gonzalo said, created a spin-off company to exploit this commercially, where we combine the structure of the receptor with docking and with estimations of binding affinity that I will tell you later, but also in the context of collaborations where we have the chemistry and the pharmacology. The next step is then to dock the compounds and see how strong they bind, the candidate compounds. And for this, first you do some docking, and then you question, okay, is this compound that binds the receptor binding stronger than this analog, where only small chemical change has happened? Which one binds stronger? And to answer this question, you better don't use a kind of like a docking program with the associated scoring function that gives you an estimation in a couple of seconds, because this will be a very approximate answer. It's okay to filter like thousands, millions of compounds, but it's not okay to do the fine-grained filtering. Now, what we do is we have developed a pipeline based on a well-known protocol called free energy perturbation. I will not get into the details here, but it uses molecular simulations to simulate the process, not the process of binding of the receptor from water to the receptor, but you compare two ligands and then you do the simulations here. And long story short, these simulations take around 2,000 to 3,000 CPU hours. Okay, so we consume a lot of computational power to do this, but they are ready in a couple of hours linear time per receptor, per ligand. And what we have is a protocol that automates this for a series of ligands. Let's say that you have a database of millions of compounds, you screen in a virtual screen, and then you have like the top 1,000 compounds. You can apply this protocol for these top 1,000 compounds to really give an accurate estimation at the experimental uncertainty of around 1 kcal or 10-fold binding affinity to run the compounds and then go back to your chemistry collaborators or to the company that you bought the compounds from and then extract the ones that you're interested. And this is fully automated in our pipeline. Now, we can do the same, but instead of studying comparing chemical compounds, we can compare variations in the protein. So we can do in silico mutagenesis. And again, the protocol is automated. It takes around 2 to 4 linear hours and around, this consumption of computing time. And the idea is the same. We can see the effect of folding the protein or protein stability, if you want the protein more stable or less, if the mutation is affecting one conformation more than the other, or types of questions that you can do with site-directed mutagenesis or pathogenic mutations you can do with this simulation engine. We have one paper that was accepted this week where we benchmark this with more than 1,000 mutations on 10 different systems, and we perform, you don't see it here, but better than a commercial solution, which is the license is incredibly expensive. We are also using machine learning, and we're doing the same kind of like question with a different approach. So the protein is represented also 3D, but here we do a graphical neural network, and we compute the properties of the wild type and the mutant side chain, and this is much, much faster. So we use it to filter the same way as we do dock compounds and then do accurate simulations to see how they bind, we can filter the effect of mutations and then fine-tune the effect of those with the simulations afterwards. This is the matter of the thesis that I'm co-supervising in Uppsala now. Now, how do we apply this to design new molecules for cancer immunotherapy? I will tell you how we have progress on this side. Okay, in this audience, it's no need to explain the tumor microenvironment, where apart of cancer cells, you have many others, but we focus on the connection between the cancer and the immune cells because we want to develop new strategies in cancer immunotherapy. So the first strategies, it was mentioned before, was monoclonal antibodies. For example, from the first one, atezolizumab, that targets PD-L1, PD-1. But we know that even if they are targeted, they present a lot of problems. High mortality, low response, etc. Now, there are many other communication pathways that we can target with monoclonal antibodies, but also there are some of the responses of the immune system, the modulators of the responses of the immune system to cancer that we can target, in this case, with small molecules. And the same as the advantages that was presented before with CDK4 and 6 inhibitors, small molecules present all these advantages that they are basically the ones that the pharma industry likes to work with. They are cheaper, they are more safe, etc. Now, we focus on, I said it before, the antagonists or blockers of adenosine receptors, because the expression of adenosine is increased in the tumor microenvironment, and this leads to a repression of the response of the immune system. So we want to block this repression, we want to block adenosine receptors. In particular, there are several A2A adenosine receptors, A2A is a subtype, that are in clinical trials, but there are very few A2B. There are some 2 or 3 A2B, A2A dual antagonists, and we are targeting this A2B receptor. Why? Because it is a low expression receptor in physiological conditions, but is hyper expressed in pathological conditions. So it gives you a selectivity framework. Now we put our computational magnifying glasses here, and this is the end result. That was the end result in 2022 of the project. We continue working on it, but we got some molecules which are even more effective than some of the molecules in clinical trials in reducing the growth of the tumor and rescuing the NK and T cells proliferation and cytokine production related to what was before. Now, how did we reach this? Now, this is just the real thing behind the cartoon. Okay, you all are familiar with this type of graphs. We reached this, started the project in 2013 by high throughput screening. This is how most pharmaceutical projects work. And in this case, it was an academic project, but the same. We screen high throughput battery of molecules, and we see if any of them block the A2B receptor. Some of them did, and they had new chemistry. We liked them. So then we started looking. This was molecules from my chemistry collaborator in Santiago de Compostela University, Edith Sotelo. You cannot see the names here. But we started looking at how the compounds bind in our model of the receptor. We do the model, we talk the compounds, and the compounds present something which is very particular. They have stereochemistry. Means that, again, something weird with the presentation, it means that there is a chiral center here, and we have to see if one molecule we have the racemic mixture. I don't know how familiar you are with the chemistry, but the molecules can exist like this or like this, and it's the same molecule. And until you separate them, you have a 50-50. So one of them we predicted was the active and the other one was not. We still didn't have the experimental result here. But when we had the experimental result, it confirmed our calculations. We compared the binding affinity of the two species with our calculations, and we saw, well, there is a preference for this form that binds the receptor in a stereospecific form, and there is one residue in the A2B receptor which is responsible for this specificity for the A2B receptor and not the other adenosine receptors. And it was confirmed with experimental circular diachromation experiments and HPLC. We did that for a number of other molecules that we synthesized with our chemistry collaborators, synthesized over the project with our assessment. And you see that in all of them, the preference was for the stereoisomer. That was the one that adopted this shape. So to this level of fine grain, we can predict the affinity of the compounds. So we continue assessing. And I told you, we can see the question from the other side. What happens when we mutate the receptor? So we identified that there was one residue that was responsible for this specificity. It existed a leucine in A2A, but in A2B, there was a valine. And the valine allowed that the L-shaped compound could fit optimally, but not the leucine. That was a hypothesis. But we did this experimentally, and we saw that the effect was specific for these compounds of favoring the binding of the compounds in the stereospecific form, but not for the regular planar compounds. And this was actually what our calculations said. So perfect correlation between the calculations and the experiments here. We assessed this over the last 10 years to fine-tune the structural activity relationship of these molecules to come with compounds that were more optimal to bind these receptors. And we are now working on dual A2A, A2B antagonists. This is more optimization. I will not get into details, some scaffold hopping. So if you are interested in this thing called scaffold hopping, finding new chemical groups that do the same as the previous ones, come to us, we can do that. I said that we can also modulate the receptors allosterically, meaning that we have to find the allosteric sites and exploit them. And this is a content from a recent review that we published on the topic, where we kind of like saw that the number of drugs that are allosteric modulators or GPCRs are very few as compared to the orthosteric ligands. There is room for exploitation here, and small molecules are still a winner here. So we can exploit allosteric sites with small molecules, but as I said, we have to determine the binding site. This is a protocol I will not enter into detail, where with some simulations and analysis, we detect the potential allosteric binding sites. And when this loads, you see just the graph, how it comes. Doesn't matter. The thing is that you have a 3D representation of the molecule with the hotspots where you can dock the molecules. We apply this on a project with some collaborators, some of them commercial companies, where they had some molecules that were allosteric modulators of the A2A adenosine receptor. And we proposed that one of the molecules binds in one side and the other one binds in the other side, based on our calculations. And actually, this fits very well the experimental data that we have, where one of the molecules have not only the EC max reduced, but also the IC50 is shifted, which is compatible with also touching the orthosteric site, whether the other molecule binds in the G protein site far from here and only reduces the Emax. Now, we can expand this and do more complicated things, bitopic ligands, fluorescent labels for labeling the receptors, etc. We're working there. This is just describing one of these projects where we did a ligand that binds in 2 different sites, optimizing it. Simulations, I will skip this. Because I want to just go back to the purinergic signaling pathway as a target for immune therapy. This is what we are now focused working on, and I will just, well, the levels of adenosine are increased, but there are many other actors in the purinergic signaling pathway. So not only the A2B receptor or the A2A or a combination of both, but also the enzymes that metabolize ATP into adenosine can be blocked. CD39, CD73, and also the purinergic receptors that are blocked by ATP and analogs. So we have been focused on adenosine receptors, but now we want to focus on dual inhibitors of this pathway. So this is the pipeline of our founded project, where Oscar is working now, and I just introduced this because this will be the matter of his seminar when his time comes to be here presenting his particular project. Different receptors, we want to target many of them. We dock compounds to the different receptors. We combine the binding affinities predicted for one and the other. There's a pipeline we support with some experimental collaborators that are working hard in getting the cryo-EM structure of one of these receptors. We purchase the compounds, we synthesize chemical variations, we test, etc. 2 years to go for this. So, as a summary, we can modulate GPCRs, and we can model how we modulate GPCRs with computer simulations. We can assess the chemists and the pharmacologists, and we can have clinical impact. We have tools that we also create and publish, and also we give the code for free, which is on the GitHub. Anyone can download it and install it. A part of this one, for example, there is a web server for doing not only the modeling and simulation of the receptors, but also in silico mutagenesis and optimizing small molecules. We also have a pipeline to detect and exploit allosteric sites on GPCRs, and we also model enzyme inhibition. I just leave you here with some examples on projects where we have modeled the COX inhibition by non-steroidal anti-inflammatories, also for some collaboration for kinase inhibitors with the University of Granada, also for epigenetics, nicotinamide methyl transferase inhibitors, and one enzyme that we are particularly interested in, which is IRAP insulin-regulated aminopeptidase, where we have a lot of activity. This is just, last but not least, the slide of all the external collaborators before where I was before. Some of them, most of them have moved some other places, including Willem, my first PhD student in Uppsala. He's professor and now in Groningen. Eddie, who is the chemistry collaborator. Mabel Loza, who some of you know, is pharmacologist in Santiago de Compostela, or collaborators at KI, Karolinska Institute, and Oscar, who is working on this project actively as a postdoc, and other external collaborators. I think we have time for questions, so I welcome them.
Gracias, Hugo. Preguntas? Even if you want them in English, I can translate, right? So, preguntas, questions? Miguel.
I have a few questions. So very nice work and pretty cool to, and I would love to start a collaboration with you, as you know. But
Me too.
looks like something, I mean, maybe I missed it, but the modeling you apply here is based on only a single polypeptide chain, or can you apply this to multiple polypeptide chains to find molecular glues or this type of stuff?
Yeah, so we can model protein-protein interactions.
Yeah.
Indeed, when I said that we have these neural network models to study the effect of point mutations on, for example, protein stability, we have also trained one of the referees as okay, can you train it? Can you show me that it works for modeling PPIs, protein-protein interactions? And we did that, and we actually are working on a large benchmark there. We can, with AlphaFold 3 and also with other methods like this Boltz 2 now, this is another alternative method to AlphaFold. We can model protein-protein interactions. For example, I mean, when I said GPCRs and G-proteins is a protein-protein interaction, but many other, and also what PROTACs do, for example, when we model like the molecules that bind here and there, which is equivalent to a bitopic ligand in GPCRs. We can model all this with external tools, and also with our particular tools, we can fine-tune the model there.
Modeling is against a region of structure you choose, or you throw the whole structure and you run the modeling to find the dog that will bind, you know. It's not the same as focusing on 20% of the protein. I mean, I can see that when you're targeting an active site, for example, to inhibit that active site. Yeah. But when you're working with the molecular glues, PROTACs, you can find other sites that are, for example, another place in the structure that can be helpful or useful for your approach. So you can just focus on the whole structure, not in a specific sites, I guess, right?
Yes, we can model at different levels. I mean, this how I can go back to how they announced the Nobel Prize in 2013 for the multi-scale simulation. So there are different levels where you can do the simulations. You can simulate the whole biomolecular association process. You can focus on a target binding site. We do both things. When we simulate the binding affinity, we do one thing I didn't extend here, and I will not do even in the answer, but one of the reasons why we are much faster than other methods is because we only focus on the binding site. But for that, you have to define the binding site. Yeah. So the first thing is that we do a simulation of the whole system. We try to see where are the binding sites, and then we bind a molecule. If the molecule has a long-range effect, then we simulate the whole thing. So depending on the question, the technique is one or the other.
I don't know what. Yeah, it's what step do you start adding variations to the structures in order to improve, for example, the physical properties of the compound that involve uptake, half-life, and all of that? Do you consider that to the original modeling, or you apply a calculator, or do you go
That's a very, very good question. So if you are really in a pharmaceutical project, you should have this into account almost from the very beginning. If you are in a more academic project where you want molecules that just bind the receptor to characterize the receptor, you can obey that to a later stage. Sometimes we are in between. For example, with the chemists that we are collaborating, they are in a pharmaceutical, as you know, in a faculty of pharmacy, and they are very pharmacy oriented. And the projects that we are having, we consider that from the beginning. We try to see if the molecules are soluble, if the molecules are not metabolized easier. So the chemical rules that we have, we consider that. There are very quick online servers to predict these things, or the ones that we have licensed for that can help you predict this. So when you filter a database of compounds, you can use these properties to filter before proceeding to optimize.
But your models do not allow to.
No, we don't work at this level. But for example, the models of improving binding affinity, they can also be used the same protocol to improve solubility. So you can just compare, okay, how well do you, how good soluble are you water against, for example, the membrane, and then you can
metabolic clearance
or for exactly.
So your readout is either interaction or activation.
Yeah, and sometimes good binding doesn't mean good activation. So that's the other thing. When you train a model, you can directly train the model with a more phenotypic readout, and you can say, okay, we don't have a direct measure of binding affinity, but we have a direct measure of efficacy. And then we kind of like can train the model to giving molecules that are efficacious, whether they bind better or not. But when we put this magnifying glass into the molecular interactions, what we model is affinity. In many cases, there's a direct correlation between high binding affinity, better efficacy. Some cases not. I skipped some slides that we have about some line of work that we did, where we model with these accurate simulations the efficacy, meaning that a molecule can bind the receptor, but in order to activate it, it has to bind the receptor in a specific conformation. So when we model is how well do you bind the active conformation versus any other conformation. So this level of accuracy we can do as well.
And you do it for either inhibitors or agonists or activators.
Yeah. Right. Okay. In this case, for example, we have a one side project on this project that we have is to get agonists from antagonists. So you have a molecule that blocks the receptor. Now I want to make chemical modifications that makes that specifically it binds in the active conformation and activates the conformation. So you can play different. It's the more accurate is the question, the more difficult it is also for us.
Any online?
Sí. Tenemos a Fran que te da las gracias y te pide que comentes el impacto de modificaciones posttraduccionales como glicosilaciones en simulaciones de docking con proteínas de membrana y receptores.
Okay, so post-translational modifications can have an effect if the effect is direct on the binding site and it has an effect on the protein conformation, you can simulate it. If the effect of the glycosylation is on the expression of the protein, for example, this is something that we don't model. We model something that has an effect on the structure of the protein or on the protein interaction with the other partner, the other protein or the other ligand. So phosphorylation of a residue, for example, can be something that can have
but the structures you work with, they are PDB files, right? So
they can be PDB files, so they can be modeled.
But they lack the modification.
They lack the modifications if they were not present on the PDB. Some of them they have the glycosylation, so it's glycosylated.
Okay.
You can model that as well, as long as you have the parameters or you develop the parameters to your simulator to understand this. So the kind of like simulator, the molecular dynamics is normally understanding the 20 regular amino acids. If there's any post-translational modification, it's either implemented or you implement it yourself and then you consider it.
Okay.